Okay. So hi. As you may know, glioblastoma is a very deadly disease. Before I explain what glioblastoma is, I'll explain quickly what gliomas are. Glioma is a cancer of glial cells in the brain, which is divided into 4 stages by the World Health Organization, with the last stage being glioblastoma in orange. And glioblastoma is the most common and aggressive form of primary brain tumors. Next, here is an MRI image of a glioblastoma tumor in a patient, and it takes up a large portion of the brain. Currently, the standard of care for glioblastoma patients is surgical removal of the tumor, followed by combined chemo and radiotherapy. Despite such aggressive treatment methods, the survival rate of glioblastoma patients remains very poor, with the median survival being less than 1 year. In addition, the standard of care has remained relatively unchanged for the past 30 years. This is why the 5-year survival rate of glioblastoma patients under the age of 40, barely exceeds 20%, shown in blue. Clearly, there's a need to develop more effective therapies for patients with glioblastoma. Researchers have shown that obese and diabetic glioblastoma patients have worse survival as compared to their normal weight and non-diabetic counterparts. In addition, obesity is an established risk factor for type 2 diabetes, both of which have links to the insulin signaling pathway. Currently, there's no detailed mechanism linking microscale insulin signaling to macroscale properties of glioblastoma. What are these properties of glioblastoma that makes it so deadly? Here they are. There's glioblastoma growth and also glioblastoma invasion. To understand the relationship between insulin signaling and glioblastoma growth and glioblastoma invasion, we have developed a computational model to explore these interactions. And today, I'll focus on linking insulin signaling to glioblastoma growth on the left. To do this, we have a 4-part approach. First, we developed a theory of insulin signaling in glioblastoma. Second, we conducted growth of glioblastoma experiments. Third, we fitted unknown model primers. And last, we conducted sensitivity analysis on glioblastoma growth. Now, I'll explain the first part. Here is a schematic of insulin signaling in glioblastoma cells. There's 3 important factors in insulin signaling in glioblastoma. There's IGF-1, IGFBP2, and HIF1α. IGF-1 is insulin-like growth factor 1. When free IGF-1 binds to IGF-1 receptors, it promotes downstream signaling through HIF1α, which in turn promotes growth in the glioblastoma cell. And also, the next factor is IGFBP2, which is insulin-like growth factor binding protein 2. It has a strong affinity for IGF-1 as it controls the bioavailability of IGF-1. It also has a IGF-1 independent interaction through HIF1α. Increased IGFBP2 levels have been correlated with late-stage glioblastoma as compared to earlier-stage gliomas. The last factor is HIF1α, and HIF1α is hypoxia-inducible factor 1 alpha. It normally acts as an oxygen sensor, so at high oxygen levels, oxygen will bind to prohydroxylases, which will recruit von Hippel-Lindau proteins to bind to HIF1α, and HIF1α will then become ubiquitinated and then subsequently degraded. HIF1α is well correlated with malignant tumors, and using this schematic, the interaction between oxygen and HIF1α was further simplified to create this, and oxygen directly inhibits HIF1α in this case. So using this schematic, we developed equations for each of these key factors, which I'll explain now. First, IGF-1 is conserved in this system. The rate of change of IGFBP2 is dependent on the production of IGFBP2, the formation and dissociation of the complex, the degradation of IGFBP2, and the promotion by HIF1α. The rate of change of complex is dependent on the formation, the dissociation, and the degradation of the complex. the rate of change of HIF1 alpha is dependent on the activation from IGF-1, the degradation and production of HIF1 alpha, and the activation by IGFBP2. And last, the glioblastoma growth is dependent on the basal growth and the additional growth due to HIF1 alpha. Now I'll explain the growth of glioblastoma experiments. We grew U87 glioblastoma cells using the hang drop assay, and we formed spheroids. Then we measured the glioblastoma diameters of these spheroids for 6 days, and we recorded the diameters, and this is shown on the left. And on the right, this is an image of what glioblastoma spheroids look like on the right. Next, I'll talk about fitting the model parameters. So in addition to using the U87 data, we also use additional data from literature. And the one on the right shows LN229 glioblastoma cell growth. And we also use 2 additional data sets, which was the one on the left shows IGF-1 to IGFBP2 relationship, and the one on the right shows the HIF1 alpha and oxygen relationship. And using all of the data sets, potential values were generated using the genetic algorithm, and then this was then further refined using the Nelder-Mead simplex algorithm and implemented in MATLAB. Once the model, once we generated the model values, we also ran the simulations under the same conditions. And here are the model predictions, the lines of the model predictions, and showing that it is in good agreement with the in vitro data, and also here. So once the model has been fitted, we also conducted sensitivity analysis on the glioblastoma growth. First, we conducted sensitivity analysis on the initial conditions. Here, we increased the IGFBP2 and IGF-1 concentrations tenfold, and then we also decreased the oxygen levels tenfold due to its inverse relationship with HIF1 alpha. And we're showing the glioblastoma diameter on the y-axis in the log format at the 24 hours. And we found that the growth was most sensitive to oxygen conditions. And this is in agreement with in vivo glioblastoma, as they generally have a very hypoxic core. Next, in order to look at the sensitivity of the rate constants, we generated something called the sensitivity index. And this is defined by the change of the glioblastoma diameters at the final time point as compared to the modified compared to the control values. And then this is divided by the maximum glioblastoma diameter, the time of simulation, and also the multiplying factor with which the rate constants were changed. And we plotted all of the sensitivity indexes against all of the rate constants. And on the left, we can see that K8, which is responsible for the production of HIF1 alpha, was the one that had the most effect on the glioblastoma growth. And now let me give some background on why we chose 2 different cell lines. So previous research, which looked at targeting IGF-1 receptors, showed that when IGF-1 receptors were targeted, there was different levels of apoptosis. So these were the different cell lines, and these are measuring the levels of apoptosis. So this suggests that in glioblastoma cell lines, where the growth is more dependent on insulin signaling pathway, which is the LN229 cell line, and there's also glioblastoma cells where the growth is not dependent on the insulin signaling pathway, which is the U87 glioblastoma cells. So we also did a very similar study where we simulated targeting each of the interactions in our model. And we did this by setting each of the rate constants to 0 individually. So in purple, this is the control growth. This is the control growth with all of the interactions shown. And here in green, this shows the removal this link, the IGF1 to HIF1 alpha link. So this is of particular interest because most of the drugs that have been developed have looked at targeting the IGF1 receptors, and it would have the same effect in our model as removing the IGF1 to HIF1 alpha link. And this is the green line here. And here, we remove the IGFBP2 to HIF1 alpha link, and this is the link. And so you can see that removing all of them separately, there's very little change in the glioblastoma growth in the U87 cell line. So this is an agreement with the existing literature data. So when we did the same analysis on the other cell line, the LN229 cell line, which shows glioblastoma cells where the insulin signaling is not a significant factor, here we remove the same interactions, and the green line, there was a reduction in the glioblastoma growth. And in particular, the red line shows that when we remove the IGFBP2 to HIF1 alpha link, we experienced the least glioblastoma growth, and the glioblastoma volume was reduced by almost 50%, by more than 50% actually. So in this cell line, it's clear that the IGFBP2 to HIF1 alpha link is the one that really drives glioblastoma growth, and that targeting this link would be an effective means to control glioblastoma growth in these type of cells. So I've talked about how insulin signaling can link to glioblastoma growth, and in the next part of my project, I'm going to work on linking insulin signaling to glioblastoma invasion, and I'm going to show a little bit about that. So here is glioblastoma cells that were cultured on a monolayer, and once they're formed on a monolayer, they can spontaneously form a pattern structure, and then they will form glioblastoma spheroids. These images is a confocal image on the left, and this was taken at 3-minute intervals, so they move quite a bit. And 10% of these cells were transfected, actually, with GFP, and this is the same image shown on the right. So using this image, we can isolate the cells and then conduct image analysis metrics on the cells, and then using this, we can quantify the movement of glioblastoma cells and link this back to insulin signaling. So in summary, I've developed a model to link insulin signaling to glioblastoma growth. I've confirmed the interactions between IGF1, IGFBP2, and also HIF1 alpha. And we've determined that IGFBP2 to HIF1 alpha link in LN229 cell line is the one that really drives glioblastoma growth. And our studies show that if glioblastoma cell growth is dependent on insulin signaling, then targeting IGFBP2 to HIF1 alpha interaction would be a very effective way to control the growth of glioblastoma. I would like to thank my advisor, the CUTUB Lab, and also the audience for their attention.
Questions? Yes.
Your model predicts that glioblastoma growth is sensitive to oxygen concentration.
Mm-hmm.
Is it correct? So are you planning, or have you already done that? It's probably possible to test this prediction experimentally. Are you intending to do this?
When I ran the simulation, I deliberately did not target the HIF1 alpha, the production of HIF1 alpha. This is because it's very ubiquitous and it's in all of the cells. So this would not be just limited to the glioblastoma cells. That's why we tried to look at something that would be more specific to the glioblastoma cells themselves.
My question was about experimental validation of modeling.
Oh, experimentally, we would plan to use different oxygen levels to conduct insulin signaling analysis. And let me show a little bit of that. So this was what we did previously. We looked at changing the insulin concentrations, and we looked at the IGF-1 receptor expression levels. And then here, we changed the insulin, and then we also saw an increase in the HIF1α levels. So we can do something similar and change the oxygen levels and measure the glioblastoma growth and also the HIF1α expression levels.
Any further questions? Thank you.